Hello and welcome, dear students, learners, and professionals, to the very interesting and important topic on biowaivers for additional strengths. And in this video, mainly the Europe regulations will be explained. All these regulations are studied based on the guideline on the investigation of bioequivalence. This guideline is for Europe region and it is a fourth part of this video series. Other videos are also available into the playlist for biowaivers. So let's start with the video. See, mainly the biowaivers means that the in vivo bioavailability or bioequivalence studies are not required or may be waived, and these are considered unnecessary for the product filing and approval, or these are not considered as a necessary part. These are based on the some in vitro studies. If the product is eligible for the biowaivers based on and supported by some of the in vitro studies, then the biowaiver can be granted. Mainly there are four types of biowaivers, like dosage form based biowaivers. These are applied to, or these are given to the products or formulations in which the drug is available in the solubilized form, like solutions, formulations, the biowaiver is directly given. Then BCS based biowaiver, these biowaiver are given for the IR formulations containing BCS 1 and 3 drugs. Then additional strength biowaiver, these are given to the strengths other than the BE strengths. So in this case, bio study or BE study, that is called as bioequivalence study, is performed on one of the strength, and the lower strengths or other strengths are given a biowaiver. Then some of the products which contain the non-absorbable APIs or which are meant for the local action in the GI tract, these are also given as biowaivers, and these biowaivers are based on the in vitro studies. So these are the types of biowaivers. Then for additional strength biowaivers, some of the key considerations are there. See, the formulations are made in multiple strengths based on the indications and the patient population. B study is passing with one of the strength and waiver is given for other strengths than the B strength. Like your product is having three strengths, 100 mg, 50 mg and 25 mg, and the bio study or B study is done on 100 mg and it is passing, then the lower strength can be given biowaiver. And the requirements of biowaiver are the same drug concentration or label claims and strength as per the RLD. If you are making a strength other than the RLD strength, then biowaiver may be problem and it is not granted. Then acceptable bioequivalence study on the B strength. Whatever the B strength you have selected, based on the bioequivalence study of that strength, the other strength can be given as a biowaiver. Then proportional similarity of the formulations across all the strengths. That may be dose weight formula, scale up, scale down formula, or the look-alike formula. The manufacturing formula should be similar and the manufacturing process should be same. Then the acceptable in vitro dissolution testing for all the strengths as per the requirements. The factors to be considered for additional strength biowaiver are as given. Drug substance pharmacokinetics should be considered. Manufacturing sameness for all the products, all the strengths. Same dosage form and only different strength should be there. Then Q1 qualitative composition, Q2 quantitative composition, drug release or dissolution profile, and the drug release profile should be similar. See, if you are making a tablet formulation for one strength and capsule formulation for another strength, the biowaiver cannot be granted. Then strength to be investigated. That means whatever the strength you are saying as a bio strength, that is B strength, and the waiver strengths. So how that B strength is selected? So as per the guideline on the investigation of bioequivalence, as per Europe regulations, the strength should be justified based on the analytical method sensitivity, safety or tolerability of the strength in healthy subjects. Sometimes if the product is not safe for the healthy subjects in the bioequivalence study, that time patients are taken. Then API pharmacokinetics and composition similarities and the differences. And the fourth point will be in vitro dissolution similarity among all the strengths. One point you should always remember that if you are doing a bio study on one strength and you are requiring or you are applying for the other strength bioequivalence, that time the strength should be compared to the bio strength and not to the innovator. The data is submitted for innovator dissolution and test product dissolution, but if your B strength is qualifying the B criteria, then based on that strength, the other strengths are eligible or not eligible for biowaiver. That should be discussed. The strength should be the most sensitive strength to detect the bioequivalence differences. Also, the state, fasting or fed, whatever we are selecting, that should be based on the product PIL, that is reference product PIL, and the product claim. The additional strength to be investigated depends on the linearity of the composition of the different strength, then linearity of the pharmacokinetics of the drug substance, then the appropriate in vitro dissolution data, that is dissolution similarity. So linear pharmacokinetics and non-linear pharmacokinetics come into the picture, and we should understand this in a very simple way. See, if you have three products, three products means three strengths, 100 mg, 50 mg and 25 mg, and you are giving 100 mg to the patient, then with that 100 mg dose you will get a AUC, that is area under the curve. Then you administer the lower strength like 50 mg. So if your pharmacokinetics are linear, then 50 mg strength will have 50% AUC compared to the 100 mg strength AUC. Then that is called as linear pharmacokinetics. And if there are some differences in the dose adjusted mean AUC, then as per the guideline, the AUC is not more than 25% or not less than 25% when comparing the studied strengths. So the pharmacokinetics are linear if the difference in the dose adjusted mean AUC is within 25% when comparing the studied strength. And if it is out of that, then it will be considered as non-linear pharmacokinetics. So in non-linear pharmacokinetics, the AUC may be higher or AUC may be lower. Sometimes due to saturation of the receptors from GIT or absorption saturation, the lower strength has little bit higher AUC than the dose adjusted mean AUC of the higher strength. And if the higher strength is having much higher AUC, that time the AUC of the lower strength may be lower compared to the dose adjusted mean AUC. So here it is very important to consider that for linear kinetics, the dose adjusted mean AUC will be within 25% of the strength which is in consideration, and non-linear may be higher or lower than the dose compared for the AUC. Now general bioequivalence criteria as per the guideline on the investigation of bioequivalence of EMA. The products are manufactured by same manufacturing process. you should not change the manufacturing process for different strengths. Then qualitative composition of the different strength should be same. Qualitative, here word is the qualitative. That means you should not use mannitol as a diluent in one of the strength and lactose as a diluent for other strength. Then it will be out of this criteria. Then the composition of the strength are quantitatively proportional. That is, the ratio between the amount of the each excipient to the amount of active substance or substances is the same for all the strength. This simply means the requirement of the compositional similarity. Here also they have mentioned that immediate release products, coating components, if tablet is a film coated or capsule shell, then coloring agents and flavoring agents are not required to follow this rule. That means this capsule shell or coating components may be little bit different in quantity and the quality. Then appropriate in vitro dissolution is required, and this in vitro dissolution data should confirm the adequacy of weighing the additional in vivo bioequivalence testing. That means the dissolution of the lower strengths or other strength than the B should be comparable and similar to the BE strength. Then coming to the third criteria, C criteria. A and B criteria we have seen. Now it is coming to the criteria C. The composition of the strengths are quantitatively proportional. That is, the ratio between the amount of each excipient to the amount of each active ingredient should be the same. Now this criteria is very important and many of the professionals found this critical to understand. But in this session, in this video, we will understand this in a very simple way with the best examples. See, in this criteria, three important points are there. The first point is the amount of the active substances is less than 5% of the tablet core weight or the weight of the capsule content. So, if the active substance into the formulation is less than 5% of the total formulation weight, that may be the core tablet weight or the capsule fill content, that time the criteria will be relaxed. Then the amount of the different core excipient or capsule content are same for the concerned strength and only the amount of active substance is changed. Third criteria is the amount of the filler is changed to account for the change in the amount of the active substance. The amount of other core excipient or capsule content should be the same for the concerned strengths. These all are required, or these three criteria are there for the compositional similarity, and that compositional similarity is quantitative in nature, because once you have the qualitative composition similarity, manufacturing process similarity, then this criteria is applicable. We will see the examples. First is composition of the strengths are quantitatively proportional. See here I have included 250 mg strength and 500 mg strength. These examples are hypothetical and these are made only for the easy understanding. Now this tablet is 400 weight and the 500 tablet weight is 800 mg. The all excipients are similar in the percentage. That means the common blend approach is applied here for 250 mg and 500 mg. The excipient ratios are also same for these both strengths. API is considered as 1 and to the 1 these excipients are considered. So for both the strengths the ratio is same. Then if your active substance is less than 5% into the formulation, see here 18 mg and 36 mg is considered. This is 4.5%. This is also 4.5%. Here For 18 mg, the tablet weight is 400 mg. Here, for 36 mg strength, the tablet weight is 800 mg. Here, the active substance is less than 5% of the total formulation weight, but the two strengths are dose weight or dose proportional, or you may also call it as scale up scale down formulation. Here also the API to excipient ratio is same for both the strengths. And this type of formulations can easily pass the criteria for additional strength biowaiver as per the EU guideline. See here in this example, 300 mg strength is considered with tablet weight of 100 mg, and 1 mg strength is considered with a tablet weight of 98 mg. Rest all the excipients are same in mg per tablet. Excipient A, B, C, D, all these are very similar to the 3 mg strength. Only the difference of this 2 mg in API weight is reduced here, and that's why this 100 mg tablet becomes 98 mg tablet for the 1 mg strength. Now the ratio of API to excipient is 28, and for B 0.12, 0.2, 0.5. Here the one diluent ratio is changing while the others are same. This formulation of 3 mg and 1 mg can be considered as similar formula or quantitatively similar formulation, because the API is within 5% and only the API is changed. Rest all the excipients are similar. Then moving to the next composition, active substance is less than 5%. Filler amount is changed with change in API and the total weight is same. In the earlier, the tablet weight was changed. Now you can see for 4 mg tablet, the tablet weight is 100 mg. For 2 mg tablet weight is 100 mg. Here the lookalike formulation is made. So this formulation will also pass the quantitative similarity formulation. The criteria of quantitatively similar formulation will be passed for this type of composition. Here you can see the excipient B, C, D are very similar, and only the diluent, that is excipient A, is changed from 83 to 85 because of the change in the API weight. Here 4 mg, so for 4 mg it is 83, for 2 mg it is 85. Now coming to the another formulation where API combination is there. API 1 is less than 5% and API 2 is not less than 5%, and it is a combination product. Now see, the two strengths are there, 10 mg by 750 mg and 7.5 mg by 500 mg. So here the excipient A is changed based on these API weights, and other excipients B, C, D are kept very similar or in proportion to the higher strength formula. Interesting to note here in this example is API 1 is 1% and API 2 is 75% for this strength, and for other strength or lower strength API 1 is less than 5% and API 2 is not less than 5%. So as per the guideline, this type of composition will not pass the biowaiver requirement, and for this type of formulation bio study may be required to be done on both the strengths. Then coming to the another example where the API 1 and 2 both are less than 5%. The total tablet weight is same and it is a combination product. So both the API are less than 5%. then you can see here only the difference of excipient A, that is diluent, is made, and all the other excipients are kept same. So this product may pass the requirement of biowaiver. Then example of combination product, where the API 1 and 2 both are less than 5%, total weight of the tablet is different, and it is a combination product. So 10 by 20 mg and 5 by 20 mg. Only one of the API is changing and other is similar. Diluent is changed as per the change in the API 1 strength. So this type of formulation will also pass the requirement. Then layer 2 is for 200 mg and 100 mg. So these both the layers are proportional. Here 200 mg, here 100 mg weight. So these can be made in combination, and biowaiver criteria is passing for this type of combination bilayer tablet, as these both the layer are dose weight formula. But always the applicant should consider a formulation case by case basis. Sometimes based on the pharmacokinetics of the API and toxicity of the API, the different strength can be required to be bio strength and not the higher strength. So every product is specific and it should be looked in a specific way. Now coming to the pharmacokinetics. Linear pharmacokinetics. Linear if the difference in the dose adjusted mean AUC is not more than 25%. See here is AUC, here is the dose. You increase the dose, AUC will be increased in a linear fashion. That's why this is a linear kinetics. Then coming to the non-linear kinetics, AUC may be more or less. So if AUC is higher than 25%, it is also non-linear. So see here, the AUC is increasing more compared to the increase in the dose. But in second example, the AUC is decreasing with the increase in the dose. So all these both examples are not that one strength may be the highest strength in general. You take the highest strength, perform the bio studies, and take waiver for the lower additional strength. If the drug substance is highly soluble, then selection of lower strength than higher strength is also acceptable. And when that lower strength can be taken? If the highest strength cannot be administered to healthy volunteers for safety or tolerability reasons. Further, if problems of sensitivity of the analytical method preclude sufficiently precise plasma concentration measurement after single dose of the highest strength, a higher dose may be selected. See, based on the method sensitivity of the bioanalysis and based on the safety and tolerability profile of the formulation, the higher or lower strength can be taken. Also, the higher dose may be selected. That means if your higher strength is 100 mg and if there is a dose of 2 tablets at a time, that means 200 mg. So that 200 mg dose can be taken, and higher dose is also can be justified based on the safety tolerability profile and the bioanalysis sensitivity. The selected dose may be higher than the highest therapeutic dose, provided that this single dose is well tolerated in healthy volunteers and there are no absorption or solubility limitations at this dose. Then coming to the nonlinear pharmacokinetics. In nonlinear pharmacokinetics, we have seen nonlinear pharmacokinetics with more than the proportional increase in AUC and less than the proportional increase in AUC. See, in linear kinetics, there is a proportional increase in the AUC. Here in nonlinear pharmacokinetics, the AUC may be lower or higher than the proportional AUC. So if the nonlinear pharmacokinetics with more than proportional increase in AUC with increasing dose is observed, that time higher strength is required to be taken. Now same criteria can be applied to this higher strength as per the linear kinetics, like lower strength if safety reasons with higher strength, and higher strength if sensitivity problems of the analytical method with lower strength. Then non-linear pharmacokinetics with less than proportional increase in AUC with increasing dose. That time we have to perform two bioequivalence studies because the AUC increase is less than the proportional dose. So higher strength and the lower strength is required to be studied for bioequivalence, or a strength in the linear range. Suppose you have 5 strengths and higher strength is say 100 mg and lower strength is say 5 mg. But after 50 mg strength, the increase in the AUC is less than proportional. So your 50 mg will be a strength in the linear range and that will be higher strength. And based on that the lower strength is selected, which is in the linear range of the AUC. So if 5 mg is not suitable for the basis of an bioanalytical sensitivity, that time higher than the 5 mg can be selected. So like that bracket can be made of 50 mg and 10 mg and not with the bracketing of 80 mg and 5 mg. And always remember, if the pharmacokinetics are less than the proportional increase, that time two bio studies are required. Now if the non-linearity is not caused by limited solubility but it is due to the saturation of uptake of transporters, then it is sufficient to demonstrate bioequivalence at the lowest strength or the strength in the linear range. This is very important. If your higher strength is non-linear or it is not showing non-linear pharmacokinetics because of saturation of the uptake, that time lower strength or the strength which is at the linear range can be selected. Then bracketing approach, bioequivalence on the two strengths, that means highest and lowest, or extreme strength, and biowaiver for the strengths which are in between the highest and lowest of the bioequivalent strength. So this bracketing can be done. It is generally required when there is a deviation in composition similarity, non-linear absorption, and when there are multiple strengths. So if you have multiple strengths, say you have 10 number of strength, 12 number of strength, you cannot make the compositional similar strength for all these strengths. That time the bracketing approach is taken, and in this bracketing approach multiple strengths are selected for bio studies and bracketing is done with the bio strengths. Then the waiver for steady state can also be taken, like fasting and fed. If B is on the one strength in both fasting and fed, and B study only in fasting or only on fed on the other strength. So if you are considering 3-4 strengths, and if you are doing bioequivalence study on higher strength in both fasting and fed state, that time the lower strength can be given a waiver of bioequivalence study in one of the state like fasting or fed state. So that time the condition selected should be the most sensitive condition or most discriminatory condition for the formulation. Like for Europe region, fasting state is generally considered as a most sensitive state and most sensitive or discriminatory condition. Waivers can also be considered for fixed dose combinations like FDC products, as well as the waivers can be based on the guideline points for the fixed dose combinations. If you are having a bilayer tablet of 2 APIs, that time those formulations can be considered for biowaiver if the product is eligible for the guideline points. Then why the biowaiver are important? Because the cost of B study is high always, then time required for B studies is more, human exposure to the drugs under B studies is there, and it is always tried to take advantage of the proven science and IVIVC so that the cost, time and exposure to human is reduced. Then biowaivers help for the faster development and the product can reach to the market in a shorter period of time. So in this video we have seen the biowaiver meaning, different types, importance, and mainly the Europe region bioequivalence guideline in details. So if you don't understand any of the point, please rewind this video and also read the guideline and bioequivalence from the EMA. See, these guidelines are always asked in the interviews. So as a pharma professional, we should be very much well informed or well understand, or we should well understand these guidelines. So thanks for watching this video. Other videos based on the biowaivers are given into the description. So these are required to be studied, like BCS biowaiver is there, bioequivalence requirement for Europe is there, and general bioequivalence information video is also there. So thanks for watching. Please do like, share and subscribe to the Pharma Learning in Depth channel. Thank you.